Okay, class, chapter 15 is on oxidative phosphorylation. This will be is not quite the last chapter we're going to go over. But if my next class pick this up, they're going to go over the whole book. This is the last class for you guys. So let's get started. Oxidative phosphorylation. We're going to be dealing with a lot of oxidation, reduction, or redox reactions. So this is a very important part of your exam. So make sure you pay attention to that. So this is one of the main things. Oxidative phosphorylation represents the final phase of catabolism of metabolic fuels and the major source of the cell's ATP. So it differs from the conventional biochemical reaction we have focused on in the last two chapters. Now, if we take a look at oxidative phosphorylation in context, this is what you see. The reduced cofactors NADH and QH2, which are generated in the oxidative catabolism of amino acids, monosaccharides, and fatty acids, are reoxidized by molecular oxygen. The free energy of this process is conserved in a manner that powers the synthesis of ATP from ADP plus P sub I. So, let's see if we take a recap of oxidation reduction. You have A and B. A is oxidized plus B is reduced, and if it's equilibrium, you have A is reduced on the other side and B is oxidized. So in this case, what happened is we have FADH2 plus Q, that's your ubiquinone, and it give you FAD, which is oxidized, and you get ubiquinol, which is reduced. So in this case, let's talk about oxidation reduction reactions are similar to other chemical reactions, which a portion of the molecule, electrons in this case, is transferred. So this is what you see here. Now, surprisingly, this is one of the things that I'm actually going over and talking to my students in 202 is reduction potentials. So this chart right here is reduction potentials of some biological substances. So if you think back to General Chem 2, you're looking at that we're looking at certain high half reactions, and these are some of them here. So you see here we got FAD on there, we got pyruvate on there, we got a acetaldehyde, we got even ubiquinol is on here too, ubiquinone going to ubiquinol. So all of those are on there. Now we talking about this, we talking about standard reduction potentials, we talking about getting delta E, we're talking about Gibbs free energy. Now, like that, we're going to also be talking about the Nernst equation. And like delta G, the actual reduction potential depends on the actual concentration of the oxidized and the reduced species. The actual reduction potential is related to the standard reduction potential by this Nernst equation. And so you guys see everything there, and that's given to you. Now, the free energy change can be calculated from the change in the reduction potential using the Nernst equation. And so this is just the Nernst equation written another way when it involves free energy change. Now, what you see right here is an overview of mitochondrial electron transport. Now, the reduction potential of the key electron carriers are indicated here. The oxidation reduction reaction are mediated by complexes 1, 3, and 4, and they release free energy. Now, what we're going to talk about next is mitochondrial electron transport. Now, we're going to map the path of the electrons through redox groups in electron transport. Again, I say this is very important, and make sure you pay attention to this. Now, the reason why I say you need to pay attention to this is because we're going to look at cellular respiration. Now, cellular respiration, when you have the NADH and ubiquinol produced by glycolysis, the citric acid cycle, fatty acid oxidation, and other metabolic pathways are ultimately reoxidized by molecular oxygen. This is what is called cellular respiration. Now, what we're going to look at in this part is electron transport change from reduced cofactors. Now, what you see here is a model of the mitochondrial structure. This is the relative impermeable inner mitochondrial membrane, and it encloses the protein-rich matrix. The inner membrane space has an ionic composition similar to that of cytosol because the outer mitochondrial membrane is permeable to substances with masses less than about 10 kD. Now, what you see in here is images of the mitochondria. So what you see in A is the conventional electron micrograph showing the cristae as a system of planar baffles. What you see in B is the three-dimensional reconstruction of mitochondrion by electron tomography, and it shows an irregular tubular cristae. And then in C, what you see is electron micrograph of a mammalian fibroblast showing the network of tubular mitochondria that is labeled with the green fluorescent dye. And the remainder of the cytosol is delineated by microtubes, and that is with the red fluorescent dye. Now, other things that you see here is the malate aspartate shuttle system. Now, cyclosolid oxaloacetate is reduced to malate for transport in the mitochondria. Malate is then reoxidized in the matrix. The net result is the transfer of reducing equivalents from cytosol to the matrix. Mitochondrial oxalate can be exported back to the cytosol after being converted to aspartate by aminotransferase. Now, you also have mitochondrial transport systems, and what you see here is the adenine nucleotide translocates binds to either ATP or ADP and changes its conformation to release the nucleotide on the opposite side of the inner mitochondrial membrane. The transporter can therefore export ATP and import ADP. And then what else happened is in B you have a P sub I H plus symport protein permits the simultaneous movement of inorganic phosphate and a photon into the mitochondrial matrix. So that's what you're seeing here. Now, what else can be going on is that you can see complexes that transfer electrons from NADH to ubiquinol. And so this is what you see here. Now, this is the structure of bacterial complex 1. There are 16 subunits shown in different colors. The redox centers are in red, FMN, in orange, that is the iron sulfur clusters. The horizontal point is embedded in the membrane, represented by the black lines, and the arm projects into the cytosol in bacteria, or the mitochondrial matrix in the eukaryotes. Now, what you see over there in B is the arrangement of the redox centers in complex 1. The atoms are coded. C is carbon is gray, nitrogen is blue, your oxygen is red, your phosphorus orange, your iron is gold, and your sulfur is yellow. Electron flows from the groups from the upper to lower right, upper left to lower right. Now, FMN stands for flavin mononucleotide. So, what happens with that is this is a prosthetic group that resembles flavin adenine dinucleotide, FAD, but it lacks the AMP group of FAD. The transfer of the 2 electrons and 2 protons to FMN yields FMNH2. So that is what happens there. Now, also what you see is iron sulfur clusters. Now, with the iron sulfur clusters, they contain up to 8 iron atoms. The most common are the 2 iron, 2 sulfur, and the 4 iron, 4 sulfur clusters. In all cases, the iron sulfur clusters are coordinated by sulfur atoms of cysteine side chains. These prosthetic groups undergo 1 electron redox reactions. Now, here, what we're looking at is complex 1. Now, complex 1 function as 2 electrons from the water soluble NADH are transferred to the lipid soluble ubiquinone. 4 protons are translocated from the matrix into the intermembrane space. Now, with all of that, what is going on? Basically, complex 1 is handling a lot of the reactions. Now, let's see what happens next. So, reactions that contribute to the ubiquinol pool. So, we go from ubiquinone, which is a ketone, to ubiquinol, which is an alcohol. Now, what happens here is in A, the succinate dehydrogenase, that's complex 2, reaction transfer electrons to the pool of reduced ubiquinone in the inner mitochondrial membrane. The acyl-CoA dehydrogenase reaction, which is one step of fatty acid oxidation pathway, generates the ubiquinone. R represents the hydrocarbon tail of the fatty acid. And then in C, the glycerol 3-phosphate shuttle system, electrons from cytosolic NADH are used by a cytosolic glycerol 3-phosphate dehydrogenase to reduce the dihydroxyacetone phosphate to glycerol 3-phosphate. The mitochondrial enzyme embedded into the inner membrane reoxidizes the glycerol 3-phosphate, ultimately transferring the 2 electrons to the membrane ubiquinone pool. Now, what here you see is the hemi group of AB cytochrome. The planar porphyrin ring surrounds the central iron atom. Shown here is oxidized in Fe3+ state. The heme substituent groups are colored blue, differ from A and C cytosomes. The heme group of hemoglobin and myoglobin has the B structure. And this goes back to chapter 5. Now, what you also see in here is the structure of complex 3. Now, with complex 3, what you see is the backbone model. Now, 8 transmembrane helices in each monomer of the dimeric complex are contributed by cytosome B. That's light blue with the heme groups in dark blue. The iron sulfur protein green with the Fe clusters orange, and cytosome C1 peak with the heme groups purple, projecting to the intermembrane space. Now, The arrangement of the prosthetic groups. The two heme groups of cytosine B and the heme group of cytosine C1 purple, along with the iron sulfur clusters, irons in orange, provide a pathway for the electron between the ubiquinol, which is located in the membrane, and the cytosine in the inner membrane space. Alright, so the complex 3 function, what we're going to see here is that I may have to go back over the Q cycle, but the complex 3 function is here. For every 2 electrons that pass from ubiquinone to cytosine, 4 protons are translocated to the inner membrane space. And this is cytochrome C. The protein is shown as a gray transparent surface over its ribbon backbone. The heme group pink lies in a deep pocket. Cytosine transfer 1 electron at a time from complex 3 to complex 4. And then the structure of the cytosine C oxidase. This is the structure here. The 13 subunits in each monomeric half of the mammalian complex comprise of 28 transmembrane alpha helixes. And then the heme group C is carbon is in gray, nitrogen in blue, oxygen in red, and iron in gold. And the copper atoms are in brown for one half of the complex. Is shown in the space filling form. And this is a model for the cytosine C oxidase activity. And then the complex 4 function, for every 2 electrons donated by cytosine C, 2 protons are translocated to the inner membrane space. 2 protons from the matrix are also consumed in the reaction. 1/2 O2 yields H2O. The full reduction of O2 requires 4 electrons. And then what we're going to talk about next is chemiosmosis. And so with chemiosmosis, the electron controlled metabolic fuse during their oxidated now fully disposed in a reduction of O2 to H2. However, their free energy has been conserved. And so what you see here, how much energy is available, this just shows you how this is collected over the different complexes, and it shows you the standard free energy for all of them. Now, right here, what you see is the generation of a proton gradient during the oxidation reduction reaction catalyzed by mitochondrial complexes 1, 3, and 4. Protons are translocated out of the matrix into the inner membrane space. This creates an imbalance in both proton concentration and electron charge. All right, last section I think is dealing with ATP synthase, learning the structure and operation of ATP synthase. Now, the ATP synthase function as protons flow through the FO component from the inner membrane space of the matrix. The F1 component catalyzes the synthesis of ATP from ADP plus P sub I. All right, now the mechanism of proton transport by ATP synthase. When a C subunit pink binds to a proton from one side of the membrane and move away from A subunit, because of C subunits from a ring, rotation brings another C subunit towards the subunit, releases the bound proton opposite side of the membrane. In mammalian ATP synthase, one complex rotation of the C ring corresponds to the translocation of 8 protons. The gamma subunit in the center of C rings rotates along with it. Okay, the structure of F1 components of ATP synthase. The alternating alpha and beta green subunit forms a hexamer around the end of the gamma cell. This view look toward the matrix onto the top of ATP synthase structure is shown here. And then that's what you see. And then the next one shows the actual binding mechanism.